Hello, everybody. We are going to start. So I'm John Lorsch, Director of NIGMS, and it's my pleasure to welcome you to our annual Judith Greenberg Early Career Investigator Lecture. As we begin, I just wanted to make note of the fact that the lecture will be recorded. We launched this lecture series in 2016 to showcase NIGMS-funded early career investigators and the many valuable contributions they're making to biomedical research. We gear the lectures to students to give them the opportunity to hear and meet early career scientists who are doing remarkable research. Our hope is that you will be inspired to pursue careers in research in this area from hearing the lecture. This lecture will begin with a speaker, Dr. Akila Rajan, giving a 30-minute talk on her research and career path, followed by a 30-minute question and answer session. I encourage all of you to ask questions about Dr. Rajan's research, along with any questions you have about her career path. And you can submit your questions to me through the Zoom chat box or by using the mic in the room if you're here with us physically. We'll also go through the questions received via the registration form earlier. Now, as I mentioned, our speaker today is Dr. Akila Rajan. Akila is an associate professor in the Basic Sciences Division at the Fred Hutchinson Cancer Center in Seattle, Washington. Her research uses fruit flies to learn how fat cells control brain function, including how fat-sensing neural circuits maintain body weight and how high sugar diets disrupt feeding behavior and response to injury in nerve cells. She has published 14 peer-reviewed research papers and served as an ad hoc reviewer for a number of scientific journals. NIGMS has funded her research through our R35 Maximizing Investigators Research Award, otherwise known as the MIRA program, since 2017. And most recently, she is the recipient of the 2023 Neurobiology of Brain Disorders Award from the McKnight Foundation, which is very exciting. And we congratulate Akila for this really fantastic recognition. So welcome, Akila, and we look forward to your talk.
Good afternoon, everyone. Thank you so much for the kind introduction. It is an absolute honor and delight to be here to tell you about my scientific journey and that of my lab using fruit flies to investigate how the organism senses nutrient flux. So I title my talk "Lord of the Flies" because I often talk to folks and the first question they ask me is, do flies have fat? So I just wanted to emphasize that they do, and that we've been making some, pretty unexpected findings by using fruit fly fat. So my motivation towards this question of fat to brain communication, as I said in the title, is that communication plays a crucial role in coordinating biological processes, whether it's within a single organism or if it's between different organisms, actually. And that this communication can occur across various scales, and it is vital for organismal function. And when I mean organismal function, you can think of many things, but I usually think about our most primal functions, like hunger, movement, sleep, and immunity. And many things can affect this organismal function, environmental factors, and primarily we think about in my lab how nutrient status impacts physiological and behavioral outcomes. So this communication, which occurs to enable an organism have appropriate physiologic and behavioral outcomes, actually across a variety of scales, time, and distance. And so I've kind of shown some of them here. They can be organellar, cellular, neuronal, and interorgan. And the focus of my group has primarily been on interorgan communication, which is a pivotal way by which information is integrated by the brain. And this information, nutritional input, can come in a variety of ways. So directly, nutritional input can impact the brain by substances in the bloodstream. Gut-to-brain communication, as you may have heard, is one of the primary ways by this occurs. But a rather more subtle manner in which this communication occurs is by the way in which the adipocytes which are the primary energy storage organ of your body, communicate with the brain. And the brain is able to then utilize this incoming information to then control all these key behavioral outcomes for the organism. So I want to just set out an outline for the expectation for my talk today, which is, first of all, I want to provide you kind of a overview of how we even got to where we got to be able to use fruit flies as an organ for, as a model system for inter-organ communication, with a focus on adipokine biology and its conservation. A majority, or rather the rest of my talk, is going to be about a story which has taken a lot of time to come to maturity, about an unexpected role for a conserved family of nutrient-sensing proteins in adipokine biology. And my goal is to convey how, by using this model system, we can continue to make discoveries which didn't seem like easily accessible by using model systems which were previously used as a primary system for that. Very ambitiously, I was also thinking I will talk about the high sugar diet work, but I think that we can, if you have any questions, reserve that for Q&A if there are any submitted through the forum. With that, I'll get going on the introduction part of the talk and convey to you my primary finding during my postdoctoral work now almost 10 years ago, in which we found that just like in humans, where one of the primary adipokines, leptin, which is secreted from fat cells and impinges on hypothalamic circuits in the brain to control the feeding status and exercise biology of mammals, there is actually a similar adipokine which is being secreted from fat cells of the fly, which impinges on brain circuits to control feeding behavior, growth, and nutrient status of flies. At the time, this finding was extremely surprising for a couple of reasons. One is that if you really look at the sequence conservation between UNPED2 and leptin, there isn't much. It's not like people have not tried to look for a leptin-like protein in flies, but if you just did a blast for leptin, and you would not pull out UNPED2 because there's absolutely no sequence similarity. They do display very minimal structural similarities, which I hope this AlphaFold image might convey to you, which is that they do have these kind of the folds that you see, but the protein itself otherwise is very different. And the size of UNPED2 is about like 45 kilodalton, whereas leptin is 15 kilodalton. Nonetheless, both of them have similarities in ways which allowed me to discover it, which is that both UNPED2 and leptins are ligands of the JAK-STAT signaling pathway. And my training prior to starting my graduate work was in the ability of cells to communicate with each other using conserved signaling pathways. And I considered perhaps instead of looking at sequence conservation, we should think about how a ligand may engage a receptor such as the JAK-STAT receptor, which is a cytokine receptor, in triggering a cascade of signaling. And this led me to find that indeed UNPED2, which is one of the 3 ligands in the fly, and it's called 2 because there are 3 ligands. And previously, no one had considered done much investigation on UNPED2 because in flies, usually, if a gene is deleted and it is not, if it doesn't display lethality, it was considered uninteresting. But UNPED2 is viable and fertile, but when you examine it very closely, it displayed metabolic effects. So I'm going to give you some very key effects here. So one is that when we subject flies to a high sugar diet, so here we are subjecting flies to 30% more sugar in the diet, we note that the average amount of triglyceride, which is your neutral fat stores, increase with time. And at the same time, if you examine the amount of relative expression of UNPED2, it increases as well. Similarly, flies, just like us, feel hungry, and UNPED2 plays a role in this, and I'm going to walk you through this experiment. One of the primary functions of adipokines is to signal satiety. So when flies are starved overnight, as it's been done in this experiment, they experience hunger, and this can be measured in a quantitative feeding assay. Whereas if you overexpress UNPED2 specifically in the adipocytes, these flies don't experience hunger. And this is because UNPED2, just like leptin, is a satiety signal. So if you artificially produce UNPET2 when it shouldn't be produced, then it prevents the fly from sensing the environmental nutrient state. Obviously, because of this reason that leptin is a satiety signal, there's been a lot of clinical interest, especially to those who think there could be a magic bullet solution for obesity with it. But our motivation in this particular work is to leverage this unexpected conservation between humans and flies to uncover biological principles that govern how organisms adapt to nutrient flux. And perhaps these findings may provide new strategies for treatments. With that in mind, ever since I established my research group, we are focused on three broad areas outlined here. One is how are neural circuits regulated by adipokines. And the second is how the adipokine retention and secretion is controlled in adipocytes. I want to take a second to emphasize that the field has actually done, especially the mammalian biology field, has done extensive work in understanding how leptin works in the hypothalamic circuits. But surprisingly, there isn't much understood about how adipocytes control the release and regulation of adipokines based on nutrient status. And I felt this was an area with our model system we could make some significant contributions. So for the rest of this talk, I'm going to speak about this until now unpublished work, which I hope will change very soon. Work is under revision right now. And since I want to now provide some understanding of, most people think of leptin as an obesity signal. So I want to come in and say, why should we be thinking about leptin during starvation? So in this really landmark work by Jeff Flyer's group in the mid-1990s, they showed that the primary role of leptin is that when fat stores fall because of a starvation status, the amount of leptin being produced decreases, and this allows the animal to adjust its neuroendocrine axis. So a very simple experiment here was they just starved mice acutely. Within a few hours, the amount of leptin in the bloodstream goes down significantly, and they artificially repleted mice with leptin, very similar to that experiment I showed you previously, and found that the mice did not do very well during starvation. They weren't able to adjust their estrus cycle. They kept their immunity up. So all these kind of quote unquote non-essential functions were not regulated appropriately to adjust for starvation. So leptin is a signal. And similarly, UNPET2 levels drop significantly by just a qPCR here, steady state mRNA levels, when flies are starved for an extended period of time, here about 5 days. And when we overproduce UNPET2, specifically, just like I showed you when it's a satiety signal, these flies don't live very long. They die faster than the control flies. So what I'd like you to take away from this is that when the starvation occurs in an organism, the levels of leptin and UNPET2 should drop. That's a key thing that needs to occur. But we don't fully understand how is the starvation status, how is it acutely controlling the release of the adipokine? And so I said this is a long-standing project in the lab. It was originally something I started to tinker around with my staff scientist and 2 postbacks who are now both in grad school, and has now come to fruition with a very talented staff scientist and a postdoc, Kevin Kelly and Aditi, working together, and we are close to being done with this, at least the first part of this story. So we began this story by examining what happens to fly adipocytes. Adult adipocytes is what you're looking at right now in a confocal microscope, and where is actually UNPET2 present. So I think that you will, first of all, wonder why I'm even showing you this, because you can't see very much here. That's because UNPET2, which is endogenously tagged here with an HA tag, it's just not very visible in normal fat tissue. Just within 4 hours of starvation, we found that when we just starve now the same fly for about 4 hours, you can now see the adipokine. The cell fills up with adipokines. That is UNPET2 in this case, and that is quantified here. But the challenge for us was that we want to correlate this with the secretory status of the adipokine. And even after trying for a couple of years, it is not technically feasible to have a strong quantitative assay to measure UNPET2. So we took an alternative approach. We used the Drosophila S2 cell system to perform certain aspects of the study and see if it recapitulates in the adipocyte. So you are going to see me showing some Drosophila S2 cell work here. And in this particular experiment, what we did is we just transfected UNPET2 transiently, this GFP tag, into S2 cells. And then we starved this amino acid-free media for just 8 hours. Again, you can see the cell fills up. So this is a cell intrinsic process, which even occurs in culture. These are not adipocytes. These are just S2 cells, which are usually derived from embryonic hemocytes in flies. And when we refeed the cells within, again, a short period of time, you kind of, again, see it gone back to its fed state. And now here we can measure this, and it's just a signal measurement. And you can see on the fed state, you see more, and in starved state, the retention goes up, and then it goes down again. And when you perform, and now we can perform a quantitative ELISA on the media, and we can show that actually on starvation, the amount of UNPET2 in circulation in the media goes down, and on refeeding, it improves a little bit. So I want to pause here and say, when you think about starvation, especially in your grad school and undergrad, the first thing that you're taught is the one way in which you survive as a cell, a starvation process, is the process of autophagy, right? Your cells undergo this process of nutrient scavenging. And one of the key components highlighted here in green, which is called LC3, is a protein that will be the prime. Autophagy is a complex process discovered in yeast, and there are many proteins that orchestrate this. But for the interest of today's talk, I just want to keep the focus on this protein called LC3. It's called ATG8. And ATG8 has this property of being able to get to conjugate to membranes, which makes it a very special protein. And in addition, it can recruit cargo, such as the one shown here in gray. And hence, it can perform this action of recruiting cargo into the autophagosome and enabling autophagosome-lysosome fusion. However, recently, the field has also started to appreciate that in addition to roles during starvation, during basal state, these autophagy proteins take on other roles. And one of them, which has come to the forefront, is secretion, that LC3 has been recruiting proteins here to a multivesicular body and enables extracellular vesicle loading and secretion. So the question becomes, is the secretory pathway here, is ATG8 knockdown phenocopying a starvation and autophagy phenotype, or is it going to look like a secretory phenotype? And we can ask that very simply by taking our S2 cells and performing just a dsRNA-mediated knockdown of ATG8. And the first thing that showed up was that an ATG8-mediated knockdown in just fed cells was equal to starvation, right? So that it seems that the adipokine accumulates within the cell in an ATG8 knockdown. And so we hypothesized, is ATG8 then required for UNPET2 secretion? We tested this, and so the purple here is the dsRNA knockdown of ATG8, and indeed, the secretion of UNPET2 is significantly compromised compared in this one. And the GFP dsRNA is just a positive control to show you that what happens when you completely remove the GFP tag. So I've been showing you data in S2 cells, and you may question whether this is relevant in fly adipocytes, in adult adipocytes. And to test this, we performed this experiment where we knock down adipocytes, we knock down ATG8 in adipocytes. But again, a tricky thing about ATG8 is that such an essential gene that if an early knockdown occurs, we will be revealing pleiotropic effects. So we did some complex fly genetics here. So what you're looking at is a mass of fly adipocytes, adult adipocytes, but only a portion of the tissue labeled in green is a genetic mosaic knockdown of ATG8. This is called clonal analysis. And what happens here is that you can see that in this clone alone, UNPET2 levels go up. So that's in blue. And if I zoom in there, I can show you. So the clone is here in green. And you can see the amount of UNPET2 accumulated goes up significantly within the ATG8 knockdown. And that's quantified here. So you may ask whether this role of ATG8-LC3 in adipokine secretion is evolutionarily conserved. So it does seem that the fly adipocytes in their fed state require ATG8 for its release, but is this relevant to human leptin? So to test this, we used an siRNA-mediated knockdown of leptin in human primary adipocytes, and we are measuring endogenous leptin secretion. So this is a positive control leptin siRNA. And when we remove either LC3 or GABA-RAP, or both LC3 and GABA-RAP, we see a significant reduction in the secretion of leptin. And so it does seem that ATG8 controls both UNPET2 and leptin secretion. And you may now ask, how is this doing it, right? It's involved in autophagy as well as controlling secretion of this key adipokine. The field has found that ATG8 interacts with most of the proteins in eukaryotes via a conserved ATG8 interaction motif. There's a lot of information here, but what I want you to pay attention to is that there is like 10 amino acids in this motif. Out of them, the key ones are highlighted in red. They're either a tryptophan or a leucine or other such amino acids like a phenylalanine or a tyrosine. And this is just a visualization for those of you who are more structurally oriented. The tryptophan and leucine in proteins which interact with ATG8, such as P62, then lodge themselves. And so ATG8 here or LC3 is shown in this structure here in gray, lodge themselves within the hydrophobic pockets, which are in yellow and green. And this is how ATG8 non-covalently interacts with its cargoes. So we asked, this is a very like conserved motif, though there is no significant conservation between UNPET2 and leptin sequence, is there an ATG8 interaction motif in either of them? And the same structure you've seen before, I've just colored the motifs. So they do have a WXXL, UNPET2 does. And for fly aficionados here, UNPET1 or UNPET3, which are not secreted, do not have the ATG8 interaction motif. And leptin seems to have two ATG8 interaction motifs right at its N terminal. And so presence of a motif doesn't really make for anything. So we performed a CRISPR-mediated mutation to the WXXL motif in the endogenous locus of UNPET2. And again, remember, when you just look at UNPET2 HA tag in adipocytes, you usually don't see much signal. I've shown you this a few times before. But when you now make this 2 amino acid change, that's the only change between the fly on top and bottom, you see a significant increase in the amount of UNPET2 retention within cells. And that is quantified there as well to show it's very significant change. And in our S2 cell ELISAs, we find that an UNPET2 AIM mutant results in significant reduction in secretion. So bottom line, what I hope you can take away from this is that on starvation or ATG8 knockdown or an UNPET2 AIM mutation results in retention of the adipocytes within the cell, even in a fed state for the latter two. So what about human cells, right? What about human leptin? Does its AIM motifs have such a phenotype? And we perform these experiments, not in adipocytes, but HEC293T, because we can make these mutations and put them back. And so here we are looking at leptin wild type tagged with MYC, and that's shown here in green. It's a transient transfection. But I hope you'll be able to appreciate that the Golgi marker GM130 is to one corner, and leptin seems to localize very strongly there in this very polarized fashion. Whereas the leptin AIM mutation causes leptin to fill up the cell. It is localized around the GM130, but also to many other spots in the cell. And the accumulation, when you quantify it, goes up significantly. And we performed an ELISA again, but now in this ELISA, what we were able to do is perform a dosage-sensitive mutation. So there are two AIMs I mentioned. So one AIM mutation in yellow and the other in salmon, they show a reduction, but when we mutate both, it shows a very significant reduction of leptin. So very simplistically, our model then is that leptin and UNPET2 in their wild type state have an AIM motif, which interacts with ATG8 and is then released outside the cell. Whereas when you examine the AIM mutations, there is some problem with it accessing the right secretory compartment, is our hypothesis, and is unable to leave the cell. We felt like this gave us a very specific handle to do a little bit more discovery. And we performed comparative proteomics of leptin in its what we call secretion competent and secretion incompetent states. So you're looking at just a Venn diagram showing what we found is like, both of them pull down about 100 proteins, but 31 proteins was very specific to leptin wild type and did not pull out with leptin AIM. And the geo terms for those 31 enriched proteins were largely endoplasmic reticulum, specific granules, and other things. So I'll come to the next slide to show you what exactly they are. And we performed a string analysis on these interactors, and we found that while we expected ER to be a part of this, we found that the cluster 3 was enriched for proteins which are involved in extracellular exosomes. But very strangely, cluster 2 contained multiple proteins of the spliceosome complex and RNA binding proteins. Very surprising. The first reaction of Aditi when she saw this was, I can't make much of this. And so we went back into literature, and this very beautiful story published by Debnath's group had shown that when you remove ATG7 or ATG8 and look at the secretome of the cell, the proteins which are not secreted are highlighted here in red, are all the ones which seem to bind to leptin. So I know this is convoluted logic, but I want to again unpack this a little bit slowly for you. And our hypothesis is that then leptin, just like these RNA binding proteins here in blue, is interacting with LC3 and getting loaded onto this secretory vesicle pathway. The beautiful thing about their story was they identified two very specific factors, the NSMAF and SMPD3, which are required for helping ATG8 access the multivesicular body. So everything I've shown you so far for human leptin and human leptin AIM has been in HEC293T cells. So this allowed us to go and perform a very specific genetic experiments in human adipocytes, where we then knocked down NSMAF and SMPD3 and asked whether removal of these factors, which are known to cooperate with ATG8 in LC3-mediated extracellular vesicle release, are required for leptin release. So with that, we did show then that actually NSMAF and SMPD3 and ATG7, which are known to cooperate in this LDES extracellular vesicle loading, are required for leptin secretion. So in conclusion, our proposal is that ATG8 and LC3 play a bidirectional role in secretion. Previous to our work, people used to think about this as, that it's playing a role during secretion in basal state, and it's kind of removed from its role in autophagy. However, our idea was that perhaps we should think more about ATG8's primary function is in scarcity, and that its basal function is in producing or releasing cargoes, and that when it's recruited to scarcity, it is no longer available to perform its secretory role. And if indeed this were true, then the question becomes whether activation and repression of autophagy have an effect on adipokine secretion. So we inhibited autophagy in this experiment pharmacologically, and we're looking at the change in secretion of an adipokine. And if you inhibit autophagy, the amount of secretion goes up. But using taurin, if you activate autophagy, we find the amount of secretion of the adipokine goes down, suggesting that there is indeed a connection between ATG8's autophagy function and secretion. Exactly how the mechanics of that works is what we are trying to work out. But what is the hypothesis out of this experiment for me as a physiologist, which is what I'm primarily interested in? What if we did the other experiment, which is we all know already that activating autophagy will promote starvation resilience. What about making an animal actively retain its adipokine, right? Like what happens to a fly if I'm constantly making UNPET2 not leave the fly, right? We could do that experiment with our genetic tools now. This is my last experiment I'm showing. I'm going to walk you through this very slowly because I really want you to get this one experiment, which is our control here is in green. So it lives for a certain number of time on starvation food. If you overexpress UNPET2 wild type, which I've shown you previously, you're producing too much of it. This is specifically being produced in the fly fat. The flies show a sensitivity to starvation. But if you overexpress the UNPET2 AIM, and all this is done in an UNPET2 deletion background, these flies indeed show a resilience to starvation. They live longer than even the wild type fly. So this suggests to us that adipocytes retention within the cell is a way for the animal to recognize that its levels are falling, and it's an active process by which organism is able to manage flux. And we think that both LC3's role in both scarcity, which is already well understood, and surplus, which is what we are revealing here, which is surplus signaling, enable the animals to maintain this balance. And with that, I'll conclude. Our main conclusion of this study is that ATG8 controls adipokine secretion and retention, and this is contingent on the nutrient state. And so in humans, in most of us, in most lean individuals, leptin is just being produced in a basal state. Now, the application for this kind of work is likely to come when we better understand what happens in a situation where, because people have shown that in obese individuals, there's too much production of leptin and it's thought that this phenomenon of leptin resistance, too much production, causes obesity to get worse. And we could potentially now try and leverage this and better understand how is, during a very surplus state, what happens to leptin secretion in flies. But in the meanwhile, in parallel studies, two excellent postdocs in my lab have been modeling the surplus state in flies. I do not have time to talk about this, but we hope by leveraging this modeling of the surplus state, we can better understand the basic biology we've uncovered in the starved state. How is it likely to impact? How is overproduction of leptin in this high sugar diet likely to happen and go wrong, is what we hope to illuminate in the future. Which that I would like to thank all of you for your attention and patience, as I had some struggles with my slides, and very, very grateful for the funding of NIGMS. I was fortunate to get the MIRA as soon as I started my lab, and it has helped support this new line of investigation on the secretory role of ATGA. Also grateful to the other funding sources for myself and my postdoc fellows. We all stand always on the shoulders of giants, and certainly I have benefited from the amazing collegiality of the fly community, and specifically of all the stock centers and Flybase and other community resources, but also my very, very generous colleagues. I mean, I could be filling up slides for the amount of reagents I've received from many of them. I'm also thankful to the excellent shared resources facilities at my institute, funded actually by NCI largely, which allow us to perform these sophisticated proteomics, lipidomics, morphometrics experiments, which I've used throughout and just alluded to in the passing, but I wouldn't be able to do it without them. And finally, I'm very fortunate to be a part of the Basic Sciences Division, which just celebrated 40 years, and thankful to my excellent colleagues, senior colleagues, for their support as well. Thank you.
Thank you so much, Akila, for that wonderful talk. Why don't you start by telling people how you got here? So, you know, how did you get to the point where you could be leading a lab and doing all this great science? What was your path?
Thanks for that question. Actually, I don't think so much about that aspect of how I got here. But I guess one thing I would say is that I had the ability of, like, you know, I was able to be in labs in which I was just allowed to explore the system and understand it and come up with my own ideas and test it. And that, like, liberty to make those kind of just experiments that fail a lot and still be able to keep going, I think was one thing that helped me, I think, continue to pursue this kind of research. And definitely, I am a big fan of my model system. I've been working on it from like more than 20 years ago. I started working on it in India. And so I do think that the kind of sophisticated genetic tools, which people, it's not like, the beauty of the fly model system is that people are continuing to innovate on the technology side of things. So for instance, we're able to do really sophisticated feeding assays and respirometry assays on flies, which we couldn't have done, like, you know, 5 years ago. And this is not something my lab is establishing. These are folks in the fly physiology field who just make these assays and make them available to us for those of us interested in exploring the biology. So I just think that definitely encourage people to think about the model system that they choose, and and in a way we are using, though the fly is an established model, we are using it to study an aspect of biology that has not been explored in the fly, and using a model system that you usually don't use to study something, and finding a way to see if it could be useful in another context is, I think, something that helped me get here. Yeah.
And how did you get interested in flies? I mean, how did you pick those as your organism?
So I actually was thinking, I mean, by now, if I'd pursued the path that I was planning to, I would be a bioinformatician somewhere. I went to a very, like, technology-oriented undergraduate institution. But there was this very serendipitous opportunity where the only way I could get to spend time at home in summer was if I took this project, which involved working with flute fly neural circuits in this place, which was, and I encountered, I think, one of the best biologists I've met. His name is Michael Bate. He wrote the book on flies, the Drosophila book on, like, you know, Drosophila Bible, basically. And it was a very serendipitous opportunity to learn about how he was using the system to just understand how disrupting neural circuits in the embryo of the fly. The question he asked was very simple. I just had to do these experiments where I would just drive a toxin in the nervous system in different circuits in an embryo and record movies as to whether the flies can hatch or not from their shell. That gave me an understanding of how such a complex, question of when is movement established in a fetus, right? You can tackle that by using just very simple genetic tools with somebody like me who just didn't have much understanding of anything, but he just sat down and taught me the genetics. And I think that was the moment I was hooked. This was in my undergrad thesis, and I've never turned back. Yeah.
Great story. There's some interest in hearing a brief synopsis of the second part, the sugar feeding story. Can you just tell people a little bit about that?
I wish I had backloaded some slides, but I apparently didn't. But let me see. Oh, I can at least use this one slide if I can. So it was just a muted slide, I think. But I think in this story, which we just published earlier last year, was a very an effort to understand when does lipid biology, when does altered changes in lipid biology start affecting feeding behavior? I know this sounds like a simple question, but it is far from it, because first of all, we didn't even know when is high sugar diet in flies starting to quote unquote break homeostasis. So this is pretty much a study of pure characterization of the model system on high sugar diet. So this is just a graphical abstract of the paper. What it shows is that we found that flies usually display this hunger-driven feeding behavior, which I just showed you numerous times in my talk, right? Like when you starve the fly, it expresses an interest in interacting with food, which we measure here quantitatively. But we noticed that around 14 days, it took about 14 days of subjecting them to high sugar diet, they were not interested in food anymore. Like they didn't display hunger-driven feeding. And we found that very curious. And we wanted to understand why that was the case. First of all, I want to emphasize here that many people in the field perform these high sugar diets on flies, but everyone stops these assays around 7 days, right? Because it just seems long enough. You should be able to, your flies, I showed you a figure in which the fat stores go up 5-fold by 5 days of high sugar diet. So folks were, and I, again, don't have the data here, but also it kind of saturates, around 5 days, the tag stores just stop, right? 14 days, 28 days, it has the same amount of neutral lipids. So we found it very curious that no one had tried it. So it was one of those experiments that we just played around. And we found that actually, if we perform lipidomics on flies, right? So just take entire flies and then crush them up and look at the entire lipid profile. And there are 13 lipid classes that the spectrophotometer we had access to could classify. And it not only looked at triglycerides, diglycerides, and known storage lipids, but was also able to assess signaling lipids like phospholipids. And we found that only after day 14, the profile of phospholipids started looking really weird, right? It just shot up and the classes were all changed. And nobody had connected very obviously phospholipids with feeding behavior or anything to do with this. And the crux of this story is that we found that a specific enzyme required for this phosphatidylethanol amine synthesis, if we just knocked that down on just a normal fly, right, it's not being subjected to high sugar diet, it displayed the same phenotype as chronic high sugar diet. So if you, my pointer is not working, but if you looked at this, the chronic high sugar diet versus the PECT knockdown displays the same phenotype. And if we overexpress this PECT enzyme only in the fat body on a day 14 high sugar diet fed fly, it continued to display normal feeding behavior, right? So and we also found that it had insulin resistance phenotypes and all that. So bottom line, what we found was that on prolonged high sugar diet, so I want to insist on the word prolonged, beyond the point where tag stores go up, other lipid species start getting disrupted, which no one had revealed before. And turns out that it's always, with science, sometimes you find something and you go back into literature. So when we were writing up the discussion of this paper, Kevin, who was the leading author on this paper, was coming to me with, hey, do you know that there are SNPs for all these enzymes in insulin resistance diseases? So there was a connection, but you could never go, didn't take a SNP and go backwards and know whether it has a causative effect or is just correlative. Whereas here, we were able to show that phospholipid enzymes in the fat tissue specifically are able to affect a behavior systemically on hunger-driven feeding. Obviously, what are the circuits being affected? How is just phospholipid change, which is a big change, having such a specific effect in the brain is something we don't understand and we hope to figure out. But that was the high sugar diet.
Thank you. That's a great story as well. There are a number of questions about how far the similarity between what you see in the fruit flies and what's known in the humans goes. So are the neural circuits the same? You know, at what point does it maybe break down if it does break down?
This is a really good question, and I wish I'd backloaded some slides on these, but we are kind of leveraging the conservation as much as it is there, but I want to highlight some clear differences in the circuits. So one thing we showed in the story in 2012 that I published in Perimon Lab was that UNPET2 impinges on, when I said the circuits are similar, it impinges on GABAergic neurons, first order neurons in the brain, the same kind of neurons on which leptin impinges on. So these neurons in turn make contact in the human brain with another neuronal subtype, which expresses something called POMC, which is propiomelanocortin, which is a neuropeptide, right? The role of propiomelanocortin when leptin impinges on is to release, and its role is to inhibit feeding and to break down fat stores, right? But in the fly, the first order neurons below UNPET2 that it impinges on is also a neuropeptide, but in this case, it's insulin. It's not the flies don't have propiomelanocortin. So there's been a switch in the neuropeptide right there. So that's why UNPET2 flies are not fat, they are lean, because insulin's role is to actually take up sugar and store it as fat. So there is like a significant difference there. But if you forget about the downstream impact, but only look at the circuit is the same. But you just have to know how to look at the circuit and know when you should stop examining. So we don't so much look into, you know, for instance, why there's been a switch between propiomelanocortin and definitely a beautiful evolutionary question. When did the neuropeptides get switched from insulin to propiomelanocortin? However, that's where the conservation ends. So there is definitely differences in the way circuits for fat breakdown are looked up in the fly. So that's why every time we run a project, we try to go and see what is the conservation in mammals to see how much further we can push it. But sometimes even the differences are very illuminating, I think. So I'll leave it there.
That's excellent. And then there's a question about the gut microbiota and how it may influence, you know, leptin and the circuits that you see.
So I will have to think a little bit before I answer this. So certainly, you know, I can't comment exactly on whether the gut microbiota are affecting leptin signaling, but given, and I'm only talking about mammals, but given that gut microbiota seem to have such a systemic impact on so many different behaviors and phenotypes, I don't know, I saw a paper about gut microbiota and autism. I can't but think that there may be some connection. But I guess it comes to then making a fly axenic, right? Like making it have no microbiota, looking at that and observing how it's changing leptin signaling is definitely an interesting line of investigation. But I have not much to comment on it because we don't work on that.
So maybe something for a postdoc to come work on.
Excellent idea, yes.
And speaking of which, there's a question from somebody who is interested in postdoc vacancies and wants to know if you're interested in neuroscience as someone who's going to become a postdoc, how do you go about finding a lab that would be appropriate for you?
Oh my gosh, this is such a difficult question, actually. So they're interested in a neuroscience postdoc. The neuroscience field in Drosophila is so wide and deep at the same time that there's so much specialization. So I would ask that person to first of all ask like what kind of neuroscience they want to do. Is this a behavioral neuroscience, a computational neuroscience, or cellular neuroscience? And then from there, I guess, read up and see what excites them the most. And then from there, the next step would be, I always say the postdoc in my lab always come in with their own ideas and see where they can, you know, I'm sure this is the same for this postdoc. It's best if they come in with whichever lab they choose to go, see what is the interesting and novel thing the lab isn't doing, and take that, but can leverage what the lab has already built to come up with a synergistic direction. I think those postdocs seem to work out in a very productive way. So I guess I remember spending like 1.5 years just reading what I wanted to work on before I ended up doing my postdoc. So it takes some time.
Yeah, very good. Number of comments on your title. A lot of people loved your title and want to know how you came up with it. So is that just in a dream? I was just playing with words, and I know the book Lord of the Flies. So it just came up, and then I honestly have chatted with a lot of like mammalian biologists, and every time I interact with them, it's happened so often now. Like I almost feel insulted. The first question they'll ask me is, where do flies have fat? They're like, I don't even know. Yeah, what is a fat fly?
I came up with this title just to kind of make it a little provocative for people to because lard is pig fat, right? Like it has nothing to do with the fly. And so I was like, maybe this will make people think. So that's how
good. Let's see. Question, another scientific question. What role does epigenetics play in these pathways that you're looking at?
So I want to say perhaps a lot, right? And I can even give some vague, hand-wavy, evidence-based things, even for instance, for the pathways we are looking at. And it's something that Kevin Kelly in my lab is pursuing. For instance, what I didn't describe and show here is that I don't know if any of you appreciated that every time we did this accumulation thing, we saw that UNPET2 is not only accumulating the cell, but is also accumulating the nucleus of the cell. Sorry for flipping through my slides. So there is this interesting idea that at least for UNPET2, we can't speak for leptin, that there seems to be some correlation between the nutrient state and the kind of, there is a lot of it in the nucleus as well. So this was interesting to us. So when we did our pull downs to look at UNPET2 and ATGate, so I'm just talking about it from this perspective. We pulled down a lot of chromatin modifiers and stuff like that. We don't fully understand it. But ATGate itself is known to, in recent, like there have been about 3 papers, I would say, one from a fly lab and one from a mammalian lab recently published, saying that ATGate controls the ability of transcription factors to drive transcription. And so it could be that ATGate is a nutrient sensing pathway, and it is present in the nucleus. I didn't go into those details, but ATGate itself is present in both the nucleus and the cytosol. On starvation, is recruited to the cytosol by the SIR2 mediated recruitment, which is again a chromatin modifier, right? But people have been looking at it more as SIR2 acting on ATGate to move it into the cytosol. But we think that in its basal state, ATGate may be performing some roles which we fully don't understand with kind of the chromatin landscape. And it's an area ripe for investigation, I think.
Excellent. And maybe one last question. This is from, I think, a student in another country who wants to know, what is your advice for a student in another country who wants to come, work in a lab in the US?
I think it's such a specific question. So like, it's very based on what they, being an immigrant student in the US was an interesting experience for me. But one thing that really helped was identifying a lab and a program, a graduate program, which is very supportive of immigrant students, right? And this can be very variable between, because there are some programs which don't take immigrant students at all because they are supported largely by training grants. And there are, so I was fortunate, so I can only speak from my experience, that I ended up in a really wonderful institute in Baylor College of Medicine in Houston, Texas, which had a large group of students from Asia and Korea and like India, China. There was a big community of us. And labs were and the lab I joined was a very international lab as well. Like, so and this helped me understand and assimilate in the culture, scientific culture and culture of the US. And I think the first thing I would do before even identifying the question is identify labs which perhaps have the track record of supporting folks who come from other countries.
Sounds like good
Yeah.
advice. Looks like we have a live question. Excellent.
Hi, Akila. Excellent talk. I did miss the first 5 odd minutes, so I don't know if my
Okay.
question will be still valid. But so there are a lot of organisms that are resistant or resilient to starvation. What do you think? What is your hypothesis how autophagy would be playing a role in such organisms?
So, you mean, I'm thinking of which organism are you talking about?
There are hibernators, there are migratory birds. I mean, I myself worked on cave fishes.
Yeah,
so
yeah, and
cave fish are
I was just
starvation
thinking of something like
resistant,
Dictee or something like that, which. So I honestly have to, I'm just riffing here and I may be totally
And
wrong.
I mean,
So do you have any thoughts, first of all?
so I don't have thoughts. It's in a given scenario, I would actually be looking at autophagy in those organisms. But another line of thought was that, say, DGRC has so many different lines. Are there, say, among those 200 odd Drosophila lines, are there some starvation resistant versus starvation sensitive, or like more starvation resistant? And then one could see how autophagy is different, what genes are playing a role. These are just thoughts that I don't necessarily have a pinpointed question,
Yeah,
but
you just like,
probably more for
so your first
discussion
part of your
later.
question was about hibernators, right? So I didn't really connect it to the fly because it's not one. So I think I'll answer this in 2 parts, right? So one thing I'll be curious to look at is whether folks who study like, hibernating squirrels or these kind of things, I know labs do this, and we think about autophagy as a nutrient scavenging pathway, but perhaps it's triggering, I'm just making this up, triggering some kind of internal change like it does with the adipokines to turn on humoral pathways which are needed for going into hibernation, right? So your animal is starving and it needs to go into hibernation. So the first thing I would do is to ask, what are the key factors required for hibernation? And are they being any way affected by autophagy autophagy? Because it doesn't mean, like, it's been studied in yeast and then up to mammals in the context of nutrient scavenging. But there's been a beautiful review written by Douglas, Green, and Galuzzi on non-conventional autophagy. The first way it starts is like every field is cursed by its first discovery, right? The curse is that ATG proteins were found for autophagy, right? But that immediately creates a paradigm, and paradigms are beautiful, we need them, but also creates this kind of fence around our own mind, which prevent us from thinking about other possibilities. And the minute you think about other possibilities, then it becomes that, and another great Drosophila physiologist told me, every time you present evidence for going outside the paradigm, then you need to present very deep evidence. So deep evidence along those lines, but I think that would be great. As for the DGRC starvation SNPs, I'm sure if you looked it up, like tons of screens have been done on starvation resilience. Of course, it'll be great to go into those and ask whether any of them are on PET2 or these mutations. That's possible. But I know that I've seen a ton of, I can't name them off of my head, but a lot of papers with she's talking about these lines available in the Drosophila field, which have SNPs in multiple genes, which are available to the community to take and screen for whatever phenotype you want. And so the question here is, has someone screened for like starvation resilience and starvation sensitivity and these kind of things? I'm very, very sure there are screens on those things. But each time with a genetic experiment, you always get a very exciting result. Driving it down to a mechanism is what takes 7 years sometimes.
I know. I understand. Thank you.
Thank you. Well, we have the Judith Greenberg lecture, and we also have something called Judith Greenberg time, which is that every meeting needs to end about 10 minutes before the hour, and we've now passed that. So thank you so much, Akila. This was really wonderful. I think it was great to see what the work you're doing, and I'm sure you inspired a lot of people.
Thank you so much.
very
I really appreciate this opportunity.